600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
What I want to show you here is a very simple table that summarizes the key differences between DNA and RNA that we've already talked about. So for instance, DNA molecules have two polynucleotide chains. RNA molecules usually exist as single polynucleotide chains. A DNA is sugar, or the sugar that we find in DNA is a deoxyribose sugar because of the simple fact that we have a hydrogen instead of a hydroxyl group at the second prime position. RNA, on the other hand, has a ribose sugar, which doesn't have, or which has a hydroxyl group in place of that proton at the second prime position. Now, DNA uses very distinct bases: adenine, thymine, cytosine, and guanine, which are for the most part also used by RNA, except for the simple fact that RNA replaces thymine with uracil. Okay, so uracil differs from thymine in that it lacks the methyl group on its ring. Now, as far as their function is concerned, at the bottom of the table right there, DNA is responsible for storing and transferring genetic information. So the genetic instructions that comprise our genome are stored in DNA. RNA, on the other hand, is responsible for creating amino acids that acts as intermediaries or messengers between DNA and ribosomes to make proteins. So to sum it up, RNA is involved in controlling the structure of DNA. So it helps in controlling the structure and the processing. Controls the structure and the processing of DNA. And it's actually what is used to convey the instructions out of the cell into the cell cytoplasm where the proteins are actually made. So what actually happens in this case, it's a very simple cycle. You start with DNA, it's read, and we create an RNA copy, a single-stranded copy of that DNA that contains instructions. Okay, this RNA is referred to as messenger RNA, which then leaves the nucleus of the cell, where DNA is located, into the cytosol or the cytoplasm, where it then proceeds to be used as a template to produce new proteins. And what I just described to you in a nutshell is a process of transcription and translation. Okay, so the conversion of DNA into an mRNA copy is what we call transcription, and the reading of that mRNA copy to create a protein is what we call translation. So we'll talk about those two processes at length when we get there, still in this lecture series. Now,